Good day, AS Biology students. I am Dr. Ruba, your AS Biology teacher. Today we're going to start chapter number 2 of our Cambridge AS Biology syllabus. This is a long chapter, and from my experience, I find that it is better that you study this chapter in sections. We have many molecules to discuss, and I don't want you to mix them up. So I'm going to divide this into 3 parts. Today, we're going to start with part number 1, where we're going to discuss the structure of carbohydrates and lipids. So let's begin. As usual, I'd like to start with reviewing all what is written in your syllabus. In the biological molecules, also part of our syllabus is going to be practical. So Benedict's test as a semi-quantitative test, and how to test and identify sugars, non-reducing sugars. This is also going to be discussed in our practical sessions. Now, the part that we're going to discuss today are the different molecules such as carbohydrates and lipids. We will talk about different types of carbohydrates and how they are made of, what are they made of, the smaller molecules, the monomers. Much detail is going to be discussed in this chapter about carbohydrates. Also, lipids are going to be discussed. They have some similarities with carbohydrates, so we're going to discuss them as well. Then proteins, a big chunk that I suggest that you should divide this video. It will be a long video because we're going to discuss a lot. I would suggest to divide it into smaller sessions. Watch part of this video when you fully understand carbohydrates, then move on to lipids, and then move on to proteins, and then maybe water as well. All right, so it's up to you, up to your time, up to your schedule, how much time do you have at hand. But this is something that will take some of your thinking, some of your time. So I would suggest to divide it into sections. So let's begin. Now, carbohydrates, this is a very smart word actually, because it tells us right away what are these molecules made of. Carbo, obviously, makes you hear the word carbon, as you hear the beginning of this word, carbo, carbon. Yes, it is the main part of this molecule. Actually, it forms the skeleton of this molecule. And then the word hydrates. Hydrates come from hydration, from addition of water. So as if we added water to that skeleton of carbon. And yes, the formula will contain carbon, hydrogen, and oxygen, where the ratio, if we compare the ratio of hydrogen atoms to oxygen atoms in this molecule, any carbohydrate molecule, we will find that they are strictly in a ratio of 2 to 1. In any carbohydrate that we have, the ratio is always 2 hydrogens to each oxygen. So let's give an example. Well, maybe you have come across a glucose molecule where there are 6 carbons, 12 hydrogens, and 6 oxygens. So 12 and 6, double that of oxygen. So that is the main formula for all carbohydrates, CH2O, and between brackets, the whole molecule is number of times, whatever it is. If it's 6 carbons, then there will be 6 oxygens and 12 hydrogens, as we just said in the molecule of glucose, and so forth. So there are 3 basic of carbohydrate molecules according to their size. So we're going to study them according to their size. If it's a single sugar unit, a single molecule, the most basic, if it's divided into smaller pieces, then it's not a carbohydrate anymore, all right? So the mono comes from 1, and saccharide, actually saccharide comes from the word sugar, And actually, it sounds more like sukkar in Arabic. Saccharide, sukkar. So probably that's where it originated. Probably, I'm not so sure. So monosaccharide is a one molecule of sugar, all right? And the formula, the general formula is, as we just said, CH2O, and between brackets, all of that, N to the power. So disaccharides is two monosaccharides joined together, and they are joined together by a special bond. We're going to call it a glycosidic bond. Glycosidic bond. We're going to talk about how this bond is formed in a while. Polysaccharides are larger molecules made up of many subunits of monosaccharides held together to make all joined together by glycosidic bonds again to make a much bigger molecule. Now, as we move from the smaller molecules to the larger molecules, we will see some changes. Obviously, the smaller molecules are soluble in water, such as glucose, fructose. These are monomers or monosaccharides. Disaccharides that are made of two sugars, yes, they are still soluble, and actually they are also sweet in taste. Monosaccharides are sweet and disaccharides are also sweet in taste, just like sucrose, the regular sugar that you have at home. Sucrose is made up of two monosaccharides joined together. There is sucrose, there is galactose. So we're going to talk about them in a while. I usually am too excited and start explaining right away. Now, let's continue. Monosaccharides and disaccharides are known as sugars. Their names end in OSE, O's, like glucose, fructose, maltose, just many other sugars as well. They have the general formula, as we said before, CH2O, all to the between brackets and N. Triose sugars, that's a group of sugars that is made up of three carbons. We have pentose sugars that are made of five carbons, and they're relevant in ribose, RNAs and DNAs, the ribose sugar. So we will find it in ribose and deoxyribose in the molecules in DNA and RNA, which we will learn in further chapters. Finally, we are going to discuss, and in a lot of detail, the hexose sugars that have six carbons, such as fructose and glucose. They have six carbons. Okay, let's move on. Now let's begin with the structure of glucose. Glucose is one of the basic sugars, monomers. It is a sugar that holds six carbons in its structure. Do you know what do we call that sugar? It's a hexose sugar. Hex comes from the word hex, like we say hexagon and such. So it has six carbons, 12 hydrogens, and six oxygens. Now we can represent that in this formula that we call a molecular formula. C6H12O6 is the molecular formula, and it tells us how many atoms are found in that molecule, how many atoms of each kind are found in that molecule. So that's the molecular formula if you haven't studied chemistry, but if you have, then you know that by now. If you want to write it to show which atom is bonded to which, this is called a structural formula. Structural formulas, such as those, well, it can be a bit detailed like this one. And here we can see the carbons. They are, as we said before, the skeleton. They form the skeleton of that molecule. So these are the six, 1, 2, 3, 4, 5, 6 carbons over here. And each of them is binded to four, it has four bonds. Some of them are double bonds to oxygen, some of them a bond to an oxygen or an hydrogen, right? But we can make it a bit shorter, easier to write it down. So this is a way to make it a bit shorter. So the carbon is bonded to OH, where O is bonded to an H. So just a way to make it shorter. But there's even another way to write it down. This is even a bit shorter, where here the intersections, in that each of that intersection is a carbon. So if we want to count our carbons, this is carbon number 1, this is number 2. As we go down, this is 3, 4, 5, and 6. This is how carbon, or this is how the molecule looks like in the dry form of glucose. Well, what happens if it's not dry? If it's added to water, it actually rearranges. So let's see, how does that happen? So the weird thing was that the structure that we have just seen, the linear structure, is actually how glucose looks in dry form. But what if glucose in nature usually is dissolved in water? That's that it behaves differently, or it actually takes a different structure. When dissolved in water, the linear glucose molecules rearrange to form a ring structure. So they're not that linear anymore. So here we're still counting the 6 carbons that we see in the structure. And what happens is that this oxygen over here that is bonded to carbon number 5 starts to attack the carbon on number 1 carbon, forming a bond between that oxygen and that carbon, which makes the structure change into a cyclical form, or a ring structure. As you can see here, now this oxygen over here, can you see the oxygen? It is one of the points in our ring structure. So if we're counting the carbons again, now this is carbon number 1 over here. And this is important to know. We will have to memorize, not just learn, memorize the structure of glucose. So we have to know how to draw it. We have to know how to differentiate it from other types of sugars. Okay, and let's begin by counting the carbons. Okay, so we used to count it on the linear structure. Now let's count. So this is carbon number 1. Down here is carbon number 2. As we go anti-clockwise, actually this is clockwise. As we go clockwise, this is carbon number 3 going up. This is carbon number 4. 5 is carbon here, number 5. And outside the ring, hanging up there, is carbon number 6. So this is the ring structure that is formed. But actually there's something even more detailed about that. As this ring is formed, sometimes the hydrogen and OH groups are not always in the same place every time. And that creates differences in the structure. Is the 3-dimensional shape is different, or just how the OH and H are situated, it is different, and that leads to different molecules. Actually, yes, we will study in a minute how this affects and makes different molecules. So I want you to note the differences between this structure on the left-hand side and the other structure on the right-hand side. Do you notice any difference? What do you note? Let's go to the next slide and look in more detail. So can you tell the differences between, well, actually it is only one difference. So can you tell the difference between these 2 molecules? Do you want some time? Well, until you think, we called one of them alpha glucose, the one on the left-hand side, and the one on the right-hand side is called beta glucose. So do you want to pause and look for the answer? The difference is, yes, carbon number 1 over here and carbon number 1 over there. There's something different. The AH and OH groups, the hydroxyl group or the OH group, is differently situated each time. In alpha glucose, it is below the ring. So if the ring is over here, you will find the hydroxyl group or the OH group below it. But if we're talking about beta glucose, it will be above it. Okay, So that's the difference. But this doesn't help me memorize it. So what did I find? Just you find a way to memorize that because it's very important. What I did is, as we look at carbon number 1 over here and carbon 1 over here, what helps me to remember it is that in the alpha glucose, the hydrogen is situated above the ring. So the A in above helps me to remember the A in alpha. This is how I remember it. But now, please remember, this is not how we write it down when someone asks us in an exam, how did we differentiate it? We don't point to the hydrogens. We always refer to the hydroxyl groups. That's how scientists like to say it. So it doesn't matter anyway, but you have to know if you're drawing the ring and someone asks you to draw the alpha glucose, then you will draw it when carbon 1, the hydroxyl group is pointing downwards and the hydrogen is pointing upwards. So if hydrogen is above the ring, this is an alpha glucose. But what if it is below the ring? This is called beta glucose. Okay, that's what we call it. These are called isomers. They have exactly the same number of atoms, but they have different 3D arrangements. And I have a lovely 3D shape for you over here. Can you see this lovely animation? This is actually the ring in the middle. Let's look together. So my pointer is a bit slow now. So this ring in the middle, okay. Can you note the colors that this model is colored? We have 3 colors. What do you think these 3 colors represent? This is good to train your brain to imagine the atoms and this molecule in 3-dimensional shape. So of course we know that the ring holds 5 carbons and 1 oxygen. That is what makes up the ring. So if I'm asking, what does the green color represent? If I see that most of the green is in the ring, only 1 is pointing outwards. So probably this is, are you ready? Did you find the answer? Yes, it is carbon. And then the red. I note that there is a bit of red in the ring. And then there are many points, many red parts outside the ring. So that helps me to remember, well, there was an oxygen in the ring, 1 oxygen in one of the angles of the ring. So that is actually oxygen. And white or gray, however you see it, this part is the hydrogen. So this represents the hydrogen atoms. Now here comes a lovely and tricky question. Is this an alpha or a beta glucose? Oops. Yes, we need to know. We have to work our brains to find out. So let's pause a bit and look at the carbon number 1. Where is carbon number 1? That's a bit tricky, isn't it? Well, let's see. If this red part is the oxygen, then the carbon that comes just below it, right below that, this is carbon number 1. And if we look at carbon number 1 now, we have a part that is hanging downwards, that looks a bit big, has a red part and a white part attached to it. So there's an oxygen and a hydrogen over here, and it's below the ring. And the hydrogen, the part that is white, is pointing out or above the ring. So which is that? Is it an alpha or a beta glucose? Where's the hydrogen? Above? Yes, it is above. So it's an alpha glucose. Good job. This is a very important molecule. You will have to train your brain and your hand to draw it, to tell the difference between it and other molecules such as beta glucose. Let's move on. Disaccharides. Now we're going to look at how these monomers are going to join together to give us a disaccharide. 2 monosaccharides make up a disaccharide. And here we have maltose. This sugar over here is a well-known sugar. It comes from barley, and we find it when we make the drink malt. And this is why the word comes from, where it comes from malt. So it is called maltose. So maltose here is made up of two alpha glucose molecules. Here is carbon number 1 on one of them, carbon number 4 on the other one, and water was removed here. And now there's an oxygen holding these two rings together. This reaction, because water is removed, we call it a condensation reaction. And this bond, we have to memorize this. I'm so sorry that you have to, but this is how we learn. So glycosidic bond is the name of the bond that we have given to any monosaccharide joining to another monosaccharide. And this is all carbohydrates. The monomers are sugars. They're monosaccharides. So any sugar binding to any sugar, this bond will be called a glycosidic bond. And this reaction, because water is eliminated or removed, we call it a condensation reaction. And this requires the presence of an enzyme because it happens in our living cells. To help it happen at a quick pace, it is catalyzed by an enzyme. The opposite of this reaction is done by another enzyme that helps to do the opposite, exactly the opposite. Instead of removing water, addition of water helps to break these two monosaccharides apart. And we call such a reaction a hydrolysis reaction. So here is another disaccharide. It's called sucrose. And this is, again, the regular sugar that you have at home. If you add sugar to your coffee or your tea, this is sucrose. And it is made up of one alpha glucose and a fructose sugar. Okay, so let's look in detail how this condensation or this reaction, hydrolysis reaction, happens. So here we have alpha glucose and another alpha glucose, and here are carbons number 1 and carbon number 4 on the other ring, and here is what happens. An OH group on carbon number 4 is removed with the H from the hydroxyl group on carbon number 1, and here this is the water that is taken away or eliminated, and here is the, which name did we give that bond? Do you remember? You have to remember. Glycosidic bond. Okay, and the same thing happens with alpha glucose and fructose. We don't have to memorize fructose. Usually questions will give you the structure of fructose and then asks you to show how it is it is joined to alpha glucose to form sucrose. So thank God we only have alpha glucose and beta glucose to remember the structure of in detail. And obviously the opposite happens. So if we go the other way around with this arrow over here, this disaccharide is broken down and hydrolyzed by an enzyme or by another condition, but here we're talking about enzymes. So water is added. Here we have some common disaccharides that we need to know. We have to know that sucrose is a disaccharide made of alpha, glucose, and fructose. And where do we find them? We find them in phloem vessels. This is how the sugar is transported from one part of a plant to another, maybe from where it is made in the leaves to where it is going to be used in respiration in other parts of the plant, or maybe where it is going to be stored in the roots, or maybe where it's going to be used to grow new cells, such as in new leaves or in new flowers or fruits, wherever. So this is where the sugar, sucrose, is found. And actually, if you know where do we get sugar, it comes from, more primarily, from sugarcane, a plant that has a long stalk or a very long stem. And this stem is squeezed through a machine and the juice is extracted out of that stem. And The stem is actually filled with phloem vessels, so this is where we get our sugar. And when we evaporate the water from that fluid, sugar crystallizes, and this is where we get our sugar. Okay, so some information for you there. Maltose. Let's move on. So maltose is made up of alpha glucose and another alpha glucose. They're bonded together, and this is found in barley seeds. Lactose is found in milk, and it is made up of beta glucose. That one is different, and galactose, another monosaccharide. Now we're going to talk about polysaccharides. So we started with monosaccharides that has one sugar. We moved on to disaccharides that has two sugars, and now we have a big molecule. We like to call such big molecules macromolecules because they're very, very big. We're talking about thousands of monomers bonded together. And here, these long chain molecules, as we just said, they're made of many monosaccharides bonded together. And because we eliminate one water molecule when each two monosaccharides come together, so it will have a certain formula. We're going to show it in the next presentation in a moment. So the four complex carbohydrates that usually come up in biology are the starch that is primarily found in potato or maybe in rice or in maize. All right, so this is where we usually get our starch. It has a certain structure that we will study in detail. Then we're going to talk about glycogen that is found in animal cells. Much of it is found in the liver of animal cells. Cellulose is obviously found in the cell wall of plants, and also we're going to talk about the structure of that. Chitin just comes along as a friend. We're not going to talk about it a lot, but I was surprised when I was studying as a teacher that it is also a polysaccharide made of many monomers bonded together. So why not put it together? So let's move on. Here, starch is made up of many alpha glucose molecules, and together when they join together, here this is the formula. We have C6H10, not 12, O5, not 6, as if we have removed the water molecule from this formula, right? We removed two hydrogens and we removed one oxygen. So this is the general formula, and we have to memorize this as well because it comes up in many questions, such as multiple choice questions. So all of that between brackets and N is outside. So if the starch molecule is made up of 1,000 uh, 1,000 monomers bonded together, 1,000 alpha glucose bonded together. So 6,000 carbons will be found in the structure of that, and 10,000 hydrogens will be found, and 5,000 oxygens will be found in that molecule. Okay, so this is a general formula for starch. Starch is found in plant cells only. Let's stress on that. We will never find starch in an animal cell unless it's a foreign body, maybe phagocytosed by a white blood cell, for example. So this is not normally found in animal cells. We do eat starch, yes. In food, we eat starch, but we digest it, and then when we store it inside our animal cells, we store it in a different form. It's not starch anymore, and we'll learn what is that. Is it in a minute. So starch is a mixture of two polysaccharides. One of them is called amylose, and the other is called amylopectin. The starch molecules have the advantage of being compact. They don't take much space, which is very good when you want to store something. You want to store a lot of that something. So it has to be not taking up a lot of space. It should be not very reactive so that it doesn't dissolve in the water and move around and we lose it somewhere or another in the cell. So it should be insoluble. It should be not very reactive. This makes it an ideal storage molecule to be found and made inside the cell. So let's compare the two different molecules found in starch. The two molecules are amylose and amylopectin. And I have a good thing to help you remember them. There is one small word molecule. Amylose is a small word in comparison with amylopectin, isn't it? Now, they're both made of alpha glucose, but there are more details to amylopectin. So that would help us when we want to differentiate them. The alpha glucose are only bonded by one type of bond, 1,4 glycosidic bonds. Why did we call them 1,4? Because carbon number 1 over here is bonded with carbon number 4. So this is how we tell what type of bond are these two alpha glucose, how are they bonded together? Because there are other ways. Obviously, that's why we had to name it this way. Now there is another way, we call it 1,6 glycosidic bond. And if you look over here, remember the carbon that is hanging above the ring over there? This is carbon number 6. And it is bonded to carbon number 1, creating a branch. So here, instead of being one long chain, now we have another long chain looking like a branch. Okay, so this creates branches, and these branches are found every 20 to 30 glucose units. While the shape of the molecule of amylose, because as you have seen in the previous and in the coming slides, you will find that the angle between these is not a straight bond, it is a bit angular. So when they are all of them angled, it looks in the end as a spiral or a helix. We call it a helix. So the shape of the molecule is coiled into a helix, and this helix is stabilized by hydrogen bonds. If you haven't heard about hydrogen bonds, we will talk about it at the end of this chapter when we talk about water. Okay, so stay tuned. Okay. In the advantages, this coiled structure is compact for amylose and is suited for storage. Over here, the amylopectin has many branches, and this provides many terminals, meaning many ends, which is that part, these ends are the parts where enzymes can attach and start breaking this molecule. So if I have a very long chain and with only one end on one side and one end on the other side, so only two enzymes can start eating or breaking down this starch molecule or this amylose. But if it has many branches, then this adds more terminals, adds more ends. So more enzymes can take part in the breaking down of that molecule, providing many sugar glucose that is ready for maybe respiration for that animal or that plant. Okay, so that's the difference. Here we have another diagram. Here is the helical structure of amylose. So you see how spiral it looks like. This takes up very less space than if it was a straight chain. Here amylopectin, it is coiled, but it has many branches, which doesn't help us to see the coiling a little bit, but still there is a coil. So we have two bonds, as you remember, we have 1,4 glycosidic bonds and 1,6 glycosidic bonds. When we test for starch, we test using iodine solution. You must have heard about that when you were studying the O-level. Iodine can fit exactly in the space inside the coil that is in amylose. So when that happens, a complex is made, iodine starch complex we call it, and it has a color. We see it as blue-black. Okay, so this is how it looks. So you can note here that the reddish-brown iodine is not reddish-brown anymore because it has reacted with the starch, and down there it looks like a blue-black color. Now glycogen, the third carbohydrate that we want to talk about. It's a long molecule as well. It is a macromolecule, but we only find it in animal cells, such as in liver and muscle cells, and we find it in fungi cells and bacterial cells, so we will never find them in plant cells. Okay, please remember that. So they do have a similar structure to amylopectin. The only difference is that there are much more branches. Each 8 to 10 glucose units, we will find a new branch. And here you can see, so if there is a coil in the middle, and these are all the branches branching out from that coil. Wow, so many, so many. And what advantage would that add? Why do we need the presence of glycogen? Why do animals need a new molecule? Why didn't we use starch to store our food? Well, because animals are more active organisms. We have a nervous system that takes up a lot of energy to help send messages from one place to another. A lot of ATP is needed for that. We have a digestive system that will contract the muscles so that the food is digested. We have muscles to move around. Plants don't have that. So all of these things are not found in plants. Therefore, animals will need more energy to be ready at hand. And there is a lovely photo here, a electron micrograph that is taken by an electron microscope. Remember from the previous chapter? Here we have mitochondria inside the cell, and these dark spots over here are the glycogen granules. This is where the glycogen is. Very well, that's a very good idea, to store the glycogen near the place where it's going to be used. So the glycogen is going to be broken down by many enzymes at the same time, releasing a lot of glucose molecules that can easily diffuse to the nearby mitochondria to be broken down by aerobic respiration to release energy, to make ATP. So that's why we need a new molecule, a design that is fit for our needs as animals. Cellulose is another molecule, another macromolecule, and it's actually the most abundant molecule on planet Earth. Wow, that's a lot. Why? Because it is found in the plant cell walls. We have a lot of plants on planet Earth. It is a polysaccharide, and it is made up of beta-glucose molecules bonded together by 1,4-glycosidic linkages. And they are arranged in such a way. I would like to move to the next slide to show you, but here you can have a look at it. So here is one beta glucose. Can you see the oxygen over here? Okay, it's a very small place to look at, but let's look in the next slide. So here are the beta glucose molecules held each is held with a bond. What type of bond? Who remembers? A glycosidic bond, yes, these are glycosidic bonds. And then together these long chains make the cellulose molecules that together make up the microfibril, and a group of microfibrils make a fiber of cellulose. And many of these fibers are found in the cell wall. Cellulose fibers are arranged like threads in the fabric. So you will find some of them in this arrangement, another in an opposite arrangement, another in an opposite arrangement. So they look like threads in a fabric running in different directions to give the cell wall their high tensile strength. This helps the plants because plants don't have a skeleton like animals do. So they need to have something to keep their form. And that is the job of cellulose and other things that we're going to study, other molecules that help to give the plant strength and rigidity. Other molecules form a glue-like matrix around the fibers that further increases the strength of the cell wall. Over here, we have an I meant to say beta glucose, sorry. Yes, this is the first molecule that we will learn, and actually the last molecule that is made up of beta glucose. Here you can see the oxygen And if we count, this is carbon number 1, 2, 3, 4, 5, and the one up there is 6. Here is the same thing, but what happens is when they are bonded together, this one will invert itself. It will flip upside down. And now the oxygen is down here. And now carbon number 1 is here, carbon 2, 3, and then this is carbon 4. So again, there will be a bond formed between 1 and 4 carbons on these two rings. Hydrogen will be removed from one hydroxyl group and the hydroxyl group will be removed. So water will be removed from this molecule. Remember what we call this reaction? Condensation reaction. Water is going to be removed. And here, this forms a glycosidic bond. We call it a 1,4 glycosidic bond, but this is a beta 1,4 glycosidic bond, okay, because it's made between two beta glucose molecules. This gives us a straight chain, not a helical chain like that found in amylose and amylopectin and in glycogen. So this is going to be a straight chain. And other straight chains nearby will form hydrogen bonds. This is an oxygen atom, and it will form a hydrogen bond with the hydrogens in this part over here, the carbon number 6, and so forth. So this makes these lines or these chains line next to one another, and they, as if they have a glue holding them to each other, which helps to give strength to this molecule. That's why it has a structural role, not a storage molecule. Cellulose is not for storage, it's for giving structure and strength. Okay? This is a lovely table that I found in one of the books that summarizes all what we have just studied. Cellulose, starch, and glycogen. And starch with its two different molecules, amylose and amylopectin. Where do we find these? We find all of them in plants except for glycogen. It is now not found in plants. It's only found in animals or bacteria or fungi cells. So cellulose and starch is strictly for plant cells. The subunits that, or the monomers that make up this molecule in cellulose, it is the only one that is made up of beta glucose, while starch and glycogen are all made from alpha glucose. The bonds, the glycosidic bonds, are 1,4 in cellulose, in amylose, in amylopectin, and in glycogen. But amylopectin and glycogen have extra bonds. We call them 1,6. Remember, these are the ones that are responsible for the branches. Yeah? And do we have branches? We just spoke about branches. No, there are none in cellulose and amylose, but there are many in amylopectin and even more in glycogen granules. And then this is just a diagram showing how these monomers are held together. Note that carbon number 6 is this one, and here it is down. So this shows us the inversion that happens in the structure of cellulose when these beta-glucose molecules come together, while here all the 6 carbons are facing in the same direction in amylose, in amylopectin, and in glycogen. So, and this is the general shape that we have. These are the straight chains in cellulose. This is the helical structure in amylose. This is the branch structure that we see in amylopectin and the more extensive branching that we see in glycogen.